Hello, this is Cell Culture Technologies Week 7 live session. This course is offered by NPTEL. The course instructor is Professor Manak Das from IIT Kanpur, and I'm Sumati Raviraj from University of Hyderabad. In week 7, the syllabus consists of fluorescence assisted cell sorting, condition for regenerated cells, introduction of skeletal muscle cell culture, and cardiac muscle cell culture. So there would have been many videos on this, and let's see the assignment for this week. So since the assignment was mostly based on fluorescence assisted cell sorting and it is a very important major tool, let's have a brief introduction about this technique. So fluorescence activated cell sorting or fluorescent assisted cell sorting, it can be called either ways. It is fluorescence activated or assisted. That means depending on the fluorescence that the cell or the tagged molecule on the cell, the signal of the fluorescence is used for detection of different cells in a mixture of cells. So each cell will have a specific marker. So based on this marker, there will be special tags associated with it based on which the uh, cell sorting will be done. So fluorescence activated cell sorting is a specialized type of flow cytometry. It provides a method for sorting a heterogeneous mixture of biological cells into two or more containers, one cell at a time, based upon the specific light scattering and fluorescent characteristics of each cells. So this means that, like I said, it resolves or it separates a particular type of cells from a group or from a mixture of different types of cells, which is nothing but a heterogeneous mixture. So a heterogeneous population of cells, when you want to isolate one particular cells, like if you have blood plasma cells and you want to isolate only the T lymphocytes, so you will give a characteristic marker of T lymphocytes, a CD marker or anything that is specific to that cell, and we will tag that with a fluorescent tag. So based on the scattering of the fluorescence of the specific light and the fluorescent characteristics, the fax machine will separate the cells, and you can also separate the cells based on the size and the granularity also. So it's based on different scattering of light, as it has been mentioned. So it can be the side scattering or forward scattering. So a heterogeneous mixture of biological cells into two or more containers. So along with detecting it as that cell, it will also sort that cell in a particular container. And this will be done one cell at a time. So single cells is very important. So the sample, when you prepare for fax, you should be very skillful and you should be very attentive to make the cells not in a clumped form. So if you have clumps, then obviously your scattering will change. It will the size of it will be bigger, and if it's a mixed population, then your different types of cells will be clumped together. So then you cannot separate the individual cell that you want to. So the sample preparation also needs to be very rigidly done, very skillfully and very particularly done, so that you have every cell in isolated form in your sample or in your media, uh, so that clumping of the cells do not occur, so that individual cells can be easily separated. So because it is one cell at a time. It is a useful scientific instrument as it provides fast, objective and quantitative recording of fluorescent signals. So it's fast because uh, the cells will be passing through it continuously, and it will simultaneously detect it, qualitatively, and also it will have a quantitative detail about it in the given mixture, that how many cells are of a specific type, and then you will have the intensity of the signals also can be measured. So it's both quantitative recording from the individual cells, as well as physical separation of the cells of particular interest. So not only detecting and identifying, like I said, it will by itself, the instrument will physically separate the cells and give you a container in which you will have the cell of your interest only. Next is, it is a technique used to detect, measure physical and chemical characteristics of a population of cells or particles. So, this FACS is a technique which is done by flow cytometry. So, the method is flow cytometry. So, here FACS is particularly for cells. So when you do flow cytometry, you can do it for cells, and you can as well as do it for different particles also, if you know some specific antigenic or some identifying markers. So you can use it for different molecules and different particles, proteins, etc., or some organelles also, like for mitochondria or some lysosomes also you can use flow cytometry. In this process, a sample containing cells or particles is suspended in a fluid and injected into the flow cytometry instrument. So like in FACS, where you prepare your cell sample, so not only the cells, you can also use different mixture of organelles, or different mixture of proteins, or whatever you want to separate the different particles. You have to suspend it in a fluid so that each cell or each particle is different from each other, separate from each other, and this will be injected into the flow cytometry instrument. So this suspended sample will flow, and it will the flow rate also will be like one cell at a time, and at which the light or the laser beam will hit. So the suspension is always a fluid suspension, and it has to be neatly resuspended so that it will have single cells. Then the sample is focused to ideally flow one cell at a time through a laser beam, where the light scattered is characteristic to the cells and their components. So FACS is fluorescent activated, so you can also do it with just the laser beam based on the characteristic scattering of the light of different types of cells. So you can even use dyes like propylium iodide or annexin, etc. So the major principle is how the light scatters depending on the type of cell. So when your sample is injected, it will flow one by one. When you have your cells, it will flow. Ideally, it should be like that. That is why you need to have individual cells in your sample itself. So you should not have a clump of cells. So if you have individual cells, ideally it will flow only one cell at a time, on which the laser beam will be hit, and then the light that is scattered from the cell will have a different scattering pattern which will be detected. Based on that, then the cell will be separated if it is a cell of your interest, or if it is a different cell. So cells are often labeled with fluorescent markers, so light is absorbed. Cells are often labeled with fluorescent markers, so light is absorbed. The fluorescent will always have a excitation and emission, and then that is how the signal is emitted. So when the laser beam hits, it will absorb a certain wavelength of light depending on the type of fluorophore, whether it is 488, 594, 561, etc. So based on the wavelength, it will absorb a particular based on the wavelength of the fluorescent, it will absorb a particular wavelength of light, and then its emission will be at a particular wavelength. So the light that is absorbed and then emitted in a band of wavelengths. Tens of thousands of cells can be quickly examined, and the data gathered are processed by a computer. So when in a suspension, you can have thousands of cells, tens and thousands of cells. So all these cells will be very quickly scattered, because laser scattering, hitting, emission, excitation is all very fast. So it will quickly process your sample, and immediately the computer that has been attached with the instrument will immediately process and give you the facts graph. Okay. So tens and thousands of cells can be quickly examined, and the data gathered are processed by a computer. Working. The three main components of flow cytometer are fluidics, optics, and electronics. The fluidic system of a flow cytometer is responsible for transporting the sample from the sample tube to the flow cell. So there are different parts of a flow cytometer. So the main components of this is a fluidics, in which your sample flows, and then the optics, your laser, etc., and then your electronics, your system associated with it for separation, and then your detector, your processor, and then also your computer. So in your fluidics of a flow cytometer, it will transport your sample. So your sample is a fluid, so your sample will be transported in a sample tube where you inject it, and then it will flow your sample one cell at a time, and then through the flow cell and past the laser, the sample is either sorted in the case of cell sorters or transported to waste. So like I said, in a mixture of cell, if you need only one particular type of cell, you will have one tag. So your laser, after hitting each cell one by one, it will detect, depending on the scattering of the light. If this is the fluorescent cell, then it will collect it in a tube. If the cell is not fluorescent, or if the cell is not your cell of interest which you need because you do not tag it, so then that cell will go into waste. That is how it will separate it. So whatever you require, you will get it in a separate tube or container, and whatever is not required will go into waste container. The components of the optical system include excitation light sources, lenses, filters that are used to collect and move the light around the instrument, and the detection system that generates the photocurrent. So your optical system is basically like your microscope system, where you use your detection of your light signal or your fluorescent signal. So for this, first you here your tag is your fluorescent tag. So for that, you have to first have an excitation light, that is nothing but your laser beam. So your laser beam will excite your fluorescent. So your laser light source is first component of your optical system. Then the excited light will be after excitation, the fluorescent dye or your fluorophore will emit a light in a particular wavelength. So this emission has to be now collected. So your emission will be collected, and it will be detected using your lenses and your filters, particular filters for different wavelengths, and then it will be detected by a photo detector or a photo multiplier, which will convert this into a photo current. Then comes your electronics. So the electronics are the brains of the flow cytometer. So till now it is simple. You have your sample, that is your fluidic system, that will pass your sample, and then you have your optical system where you have light, your lens, your filter. So it's based on your excitation and emission of your fluorophore. Now whatever signal it has gotten, it has to now analyze it. So for this you need electronics, and here you have from the photocurrent which is generated by the detector, it will be processed. So the electronics are the brains of the flow cytometer. Here the photocurrent from the detector is digitized and processed to be saved for subsequent analysis. So after that, you can further have other detailed analysis also once you get your flow cytometry or your facts graph based on the gating and based on your forward and side scattering. So this is how it is. So the three main systems are your fluidics, your optics, and your electronics. The fluidics is your transporting for the transportation of your sample, that is your in your sample tube. It will flow your sample one cell at a time, and then you have your optical system, which includes your source light, your lens, and the filters, and then also the detector, which will uh, convert the signal into a photocurrent. So now your photocurrent has to be digitalized and it has to be analyzed. That will be done by your electronics. So then you can further do your particular analysis, if it is depending on your experiment or if it is just to separate your cells, isolate your particular type of cells. So this is a picture to just demonstrate how this works. So this is your sample. This is your sample. So here you have a mixture of cells. So around this you need only one type of cells which express a particular type of protein or a particular type of antigen in this case, because we use antibodies to detect it. So now you have these cells in which is your target cells, your green, and then it won't be green in color, it's just to differentiate for our purpose of understanding. These are your cells. This is type A cells, this is type B cells, but all of them will not be having any dye or color in your sample initially. Whichever cells you want to label, you have to know the marker for it. So then if you have a marker, you will make a antibody with a fluorophore tagged to it against that particular marker of your cell of interest. So then you have your antibody labeled with RFP. Now when you mix your fluorophore with your sample, then your fluorophore tags will go and attach to your target of interest or your cell of interest. Now the non-target cells will not have this antibody attached to it. So now your non-target cells will not have any fluorescence, but your target cells which have now been bound by the antibodies which are tagged by fluorophores will emit fluorescence when hit by a source light of a particular wavelength. Okay, so now in this mixture you will load it into your flow cytometry system. Now your sample of mixtured cells goes into the system, your sample tube, and now your sample tube will send the cells one by one through a tube-like structure, and then once it has been at a flow rate of one cell at a time, your optics or your laser system will act on each cell. So whatever cells are not tagged with your fluorophores will go into waste, and whatever cells are tagged with your fluorophore will emit fluorescence, and this fluorescence will be detected by a fluorescent detector, and the light scattering will also be detected by your scatter light detector. So now based on these two, for your scattered light you have your forward and side scattering, and for your fluorescence you have your particular wavelength of emission. Based on this, now your electronics will process the information and give you the graph. So here it has been put in the same container in the picture, but your non-target cells or your non-tagged cells will go in a different container. So it will be like a uh, uh, like it will be in a way that it will split the cells into different containers. So your non-target will go into a different container, your target will go into a different container. So it is that quick that by the time your cell falls down, it will immediately capture your fluorescence, it will analyze and then identify that this is your cell and immediately transfer it into your collection container. Okay, so now let's see what else we need to know particularly about this. So cell mixture containing fluorescently labeled cells exists via nozzle. Then this is your nozzle in the previous picture. Then the laser beam strikes the droplets. Then the forward scattering detector identifies the size of the cell, and the side scattering detector identifies the granularity or fluorescence of the cell. So that is why you have to have single cells. If you have a clump of cells, even if they are the same cell, it will be detected as a different cell because of the difference in the size of it. The clump will be larger in size, so it will have a different forward scattering than the single cells. And then so based on this different size also, you can differentiate among your cells. Like if you have variation in your mixture. If your cells are very different in size from each other, then you can easily use forward scattering to separate them. And then your side scattering detects the granularity and the fluorescence of the cell. The electrode assigns positive or negative charge. So after this is done, the electrode will assign a positive or negative charge for each cell. Based on this charge, the cells will then be sent into different tubes. So the positive charged cells are drawn to a negative plate, while the negative cells are drawn to a positive plate. The separated cells are collected in a different collection tube. So this is how, this is the flow of your facts. Okay. So cell mixture containing fluorescently labeled cells exists via nozzle. Laser beam strikes the droplets. Forward scattering detector, side scattering detector. They detect size of the cell and granularity or fluorescence respectively. Then the electrode assigns a positive or negative charge. Positive charged cells will be drawn to negatively charged plates, and negatively charged cells will be drawn to positively charged plates. This is how they separate the cells into different tubes. Next is, what are the applications of this? So first, to be a cell structure, it includes cell size, your forward scattering. So you using this, you can implement it to differentiate among a mixture of different sizes of cells, and in a mixture of different sizes of particles, or based on the surface area, karyoplasmic ratio, content of DNA, RNA and proteins can be detected, because everything has a different scattering. And then your cell cycle also can be detected, because while during cell division your cells undergo different stages, so each stage can be detected in the cell in a mixture of your population, which cell is at which stage of your mitosis. And the second comes to be cell function. So this is based on the physical properties of the cell. Now based on this functioning of the cell, you can use your specific antigens, you can use your cytokines, you can use your detect your different cell activity, enzyme activity, hormone binding sites, receptor sites, etc. Then immunophenotyping. This is a method that identifies and quantifies multiple population of cells in one heterogeneous sample. This includes peripheral blood, bone marrow and lymph material. Then cell sorting. Advancement in cell sorters are geared towards the gentle isolation of cells into separate tubes. Cell cycle analysis. With flow cytometry and FACS, the cells can be analyzed and measured in all four distinct phases of entire cell cycles. That is your prophase and metaphase, anaphase and your telophase. Then your apoptosis. These two distinct types of cell death, apoptosis and necrosis. So you have two types of cell death. One is apoptosis, one is your necrosis. Both can be identified via your flow cytometric methodology and used to be determined morphologically, biochemically and molecular changes that had been occurring in dying cell, because the pathway for apoptosis is different and necrosis is different. So based on your biochemical pathway in it, you can detect these two types, difference between these two types of cell death. Then cell proliferation assays. As the cells divide, half of the original dye is passed on to each daughter cell. So when the cells take up the dye or your fluorescent, and then it divides into two cells. So half of your dye will go in one cell, half of your dye will go in another cell. So your signal will become half. That is what is used here. So by measuring the reduction of your fluorescent signal, researchers can calculate the cellular activation and proliferation. So how you use this in your experiments, you can design it in your own way. Based on the knowledge of the principle of the technique, you can design your own experiments. So this is one example for that. Then intracellular calcium flux. Flow cytometer is able to detect the flux of calcium into the cell and provide detailed measurement data. So several times during stress or during particular type of conditions, there will be increased calcium flux into the cells. So this also can be measured by flow cytometer. So till now is the introduction basic for flow cytometer. Now we go to the assignment. So since this time because of the email discrepancy or the reminder that was not sent, there were no students that has joined, so I will just go through the questions and there won't be any interaction because of absence of students. We will just go and see what the answers are. Which of these cells can be used to separate population? Which of these can be used to separate population of two cell types from a mixture? Immunopanning, fluorescent assisted cell sorting, both A and B, none of the above. So, for separation of cells from a population, you can use both immunopanning and fluorescence assisted cell sorting. So here you can see your GFP labeled cell population. Different cell populations are there. So after preparing the samples in a similar way, you can use it for different types of cells, not only your animal cells, different type of cells if you have the tags for it. Then the mixture of cells has been passing through the sample tube. You have a laser which hits each cell and detects the signal. Then based on the signal, the electrode will impart charge on it, and then positively charged cells will go in one, negatively charged cells will go in another tube, non-charged or uncharged cells will go in a different tube. So here you can see they are collected and separated in 3 different tubes. Now this you can use for your transcriptomics, proteomics, metabolomics, anything. Okay, then let's see what is immunopanning. So in an immunopanning, this is a simple overview of it. So when you have, for example, when you take an animal model, you after dissection, we optimize by pepin because you need separated cells, and then you will have trituration, and then optimized panning steps for your particular antigen or antibody, whatever you have for your cell of interest for the selection. So you will have that, and your cells will be selected using that particular antibody. So what are the benefits of this is, yeah, like in different stages of cell or for different cells where you need to isolate some primary cells and then grow it, so you can easily isolate one particular type of neurons or one particular type of cells from a tissue and then grow it as the pure culture. So that is the one of the important applications for this. So next question is, which of these is a signature for all leukocyte groups? So for all leukocyte groups, CD45 is always there. So these will all be CD45 plus. So now you can see here different markers that are present on different population of cells. So why we have to know this is, you can make use or take advantage of these specific markers on these cells to isolate and identify them. So your CD45 is present on all leukocytes. So if you have a mixture of blood cells, or if you have a mixture of cells from a sample and you want to isolate only leukocytes and all the leukocytes, excuse me, all the leukocytes, then you can use your CD45 as your marker tag, and all your leukocytes will be separated in your container. So now again within your leukocytes you want to isolate different types of cells. So then you can use combination of these markers to get your main population. So then, for example, your CD3, CD4, you have your T cells mostly, and then your CD8, your CD16. So all these, if you use a combination of markers like 45, 16, 56, you will have only NK cells. If you have a specific marker like CD62P, you will get only monocytes. So these kind of markers you can make use to separate your cells of interest. Next is a dash is a protein molecule that recognizes dash on the cell surface. So an antibody is a protein molecule that recognizes the antigen on the cell surface. So what is the difference between an antigen and antibody? So your antigen will be expressed on the cell surface. So it will have a epitopic region which determines its antigenic property, which allows it to be able to, or which allows it to be detected by an antibody or a generation of antibodies. Such regions are epitopic regions of an antigen. So you can see the red ones are the antibodies. So antibodies once generated, they will be in circulation. So they are protein molecules and they go in circulation and go and fight the complementary antigen and go and detect it. Antigen is the substance that, when introduced into a vertebrate host, provokes an immune response, leading to acquired immunity, whereas antibodies are specific proteins synthesized by the host in response to an antigen. Okay, so that is the difference between antigen and antibody. In DASH, there are two types of motor neurons. One are small in size, called DASH, and other are large in size and called DASH. So the one that is small in size are your ventral horns, and the other large in size are called okay. So one that is small in size and large in size are your small in size is your alpha, large in size is your gamma. So both of these are present in your ventral horn. So you can see here, so your alpha motor neurons and your gamma motor neurons. So your alpha are mostly extrafusal and your gamma are mostly intrafusal. That is their innervation in muscle fibers. The function of alpha motor neurons is for muscle contraction. Size of alpha motor neurons are larger and gamma motor neurons are smaller. So this is a picture depicting your spinal cord, and so these are your muscle spindle fibers. So these are your gamma and these are your alpha. Okay. Next is, in rat spinal cord at E14, only motor neurons express a particular marker. This information can be used to isolate these cells from a mixture population with other cells of the spinal cord, test the electrical activity of these cells, digest these cells, none of the above. So you can use this information like we excuse me. Yeah. So like we discussed, when you know a particular type of cell expresses a very specific marker, you can use it to isolate the cells. So the answer is, isolate these cells from a mixed culture with other cells of the spinal cord. So here we can see an example of TUJ1 that is being used here, and SHAT, and then islet 1 cells, islet 1 protein. So you can see the neurons, how you can specifically tag them and then use it for isolation. So the homeodomain factor HB9 is expressed in motor neurons at early and late stages of differentiation. So HB9 is critical for distinguishing between motor neurons from interneurons. Immunofluorescence staining can be used to extract motor neurons from E14.5 embryonic spinal cords. So it's your embryonic, E14 means your embryonic. And then the motor neurons can be characterized using neuronal markers such as TUJ1, NFL and NFM, and motor neuron specific markers such as SHAT and islet 1. To purify motor neurons, the gradient tube can be centrifuged for 15 minutes at 800 G at 22 degrees Celsius, the desired layers can be collected and transferred in a new tube for highest purity. Layer 3 can be collected. So this is like a just an example for like how this protocol works. So when after tagging and then you have your identification, you can use your flow cytometry or your immunopanning, all these type of methods, or if they are different in size and density, you can even have a density gradient isolation. So you can do your centrifugation and then you will have different layers of cells based on the density of the cells. So you can use them and easily identify your type of cell. Next, trypsin is used to dash by digesting the extracellular matrix as it is a dash. So lift the culture cells of a surface, it's a condensation enzyme. Lift the cells of a surface, proteolytic enzyme. Attach the cultured cells, condensation enzyme. Attach the cultured cells, proteolytic enzyme. So your trypsin we have discussed before also, it's a proteolytic enzyme and it is used to detach your cells from the surface. So the answer is B, lift your cells from the surface, and it's a proteolytic enzyme. So quick recap of the trypsinization method. So here you can see your tea flask in which the cells are cultured. Any cells which are adherent in nature, where you have the need for trypsin. So like your HT29 cells or your FG2 cells or your MCF cells, your monocyte cells, or your HEK cells. So anything, your adherent HEK cells. So any cell line which is adherent in nature, you need trypsinization. So after growing the cells in your media is aspirated from your plate, and then a monolayer is rinsed with 5 ml of PBS or phosphate buffer to remove the serum, and that will also be aspirated out. So your serum contains components which will neutralize your trypsin. So that is the reason we give a wash with PBS, so that the residual media is also washed away, which has remnants of serum. So now after washing off and removing the PBS, and you will add 1 ml of your trypsin. So trypsin percentage also depends on your cell type. So you can have either 0.25% or 0.05%, etc. Mostly trypsin is given in combination with EDTA, because EDTA is a calcium chelator. So it will help in activation of trypsin. Then was added and allowed to spread over the monolayer of cell lines, followed by incubation for 5 minutes. So after adding this, you have to incubate it, because the enzyme has to get activated at 37 degrees, that is its temperature of optimal activation, and then it will detach each cell by proteolytic breakdown of the proteins. After which cells are suspended in 8 ml of media to stop the action of trypsin. So again, as I said, serum neutralizes the activity of trypsin. So now to stop the proteolytic cleavage, we will add serum containing media so that trypsin will get deactivated, because increased activation of trypsin or longer time exposure to trypsin will damage the cell surface proteins and the cells also. So gently pipe it up and down to break the clumps and to observe in the microscope. So now you have to resuspend the cells very gently, so that the detached cells do not remain in clumps. They are single isolated. Like we said, we talked about this in seeding and subculturing. So when you seed in a new plate, you ideally should have single cells, so that it does not grow overlapping on each other. It should grow as a monolayer or a single layer. Then after this, we observe the cells in the microscopes to see if they are in healthy condition, not damaged, and are not clumped. So after that, the cell suspension is transferred into another tube, and then it is centrifuged for 5 minutes around 1000 to 1200 rpm. Then after which after which you will discard the supernatant and then add fresh media and then resuspend it either gently with pipette or just by shaking the tube gently and uniformly so that your cells will get dispersed in the media. And now you will seed your cells based on the number of cells required after cell counting. So this is the method for trypsinization, how you use trypsinization for subculturing. Next is, which of these is not a component of B27 supplement? Vitamin A, biotin, acutase and sodium selenite. So acutase is also a detachment agent or dissociation agent like trypsin. So that will not be in your B27 supplement. So your B27 supplement is a mixture of vitamins, proteins, fatty acids and antioxidants, enzymes that supports the survival of neuronal cells. It's a serum-free supplement that's used to support the growth and viability of central nervous system neurons. So these are the components of your B27 supplement. Biotin, DL-alpha tocopherol acetate, DL-alpha tocopherol, vitamin A, some proteins, and then BSA, like BSA, fatty acid free fraction 5, catalase, human recombinant insulin, transferrin, superoxide dismutase, etc. And other components like your corticosterone, D-galactose, ethanolamine, glutathione, L-carnitine, linoleic acid, progesterone, putrescine, sodium selenate, T3, etc. So your B27 supplement contains vitamins, proteins, fatty acids and antioxidant enzymes majorly. The purpose is to support neuronal survival in cell culture, and it can be used for growth and maintenance of neuronal stem cells and also in 3D cell culture, disease modeling and many more. Variations for these formulations are also there. So where you can have formulations without insulin or without vitamin A, depending on the experiment and your necessity of the design of your project. Next is Glutamax is dash and has dash than glutamine. So Glutamax is nothing but L-alanine, L-glutamine, and it is used because it is more stable than glutamine. So your answer is L-alanine, L-glutamine, and it is used because of its higher stability than glutamine. So stability of Glutamax is more than L-glutamine and does not break down spontaneously. L-glutamine can break down into toxic byproducts like ammonia and pyrrolidine carboxylic acid. Cell growth. Glutamax can help cells grow efficiently and yield high growth. Ammonia buildup. Glutamax can help prevent toxic ammonia buildup and that can occur with L-glutamine. Shelf life, it can have a extended shelf life in the culture media, and temperature stability remains stable at room temperature for up to 2 years and can also be stored at different temperature for convenient handling. Glutamax is a supplement that can be used in cell culture to improve the cell growth and viability and to avoid negative effects of ammonia on the cells. This is the major reason for usage of Glutamax. One is to avoid toxic effects of ammonia, which usually occurs with glutamine, and it is required for the growth and viability of cells. Next, so DASH is the supplement used for serum-free fetal hippocampal neuron culture media. So the answer is B27 supplement. So B27 was developed as a supplement to DMEM for the growth of hippocampal neurons by optimization of over 20 components of a previously published serum-free supplement called B18. So this is a more advanced and better version of B18. So B27 is a serum-free supplement that supports the growth and long-term viability of hippocampal neurons. It is a mixture of proteins, vitamins, antioxidant enzymes and fatty acids. B27 is used with a basal medium like neurobasal medium. NCS27 is also a serum-free medium supplement that is modified version of B27. So B25 is a better modified version of B18, and NCS21 is a modified version of B27. It can be used for cultivating neuronal cell types, long-term growth of hippocampal neurons, and more. The last is read the statements carefully. So this question is not theoretical, it is analytical. So this I want you all to do it on your own. So I will just give a little hint for this. So your animal origin medium. A is not manufactured using any raw material ingredients derived from an animal tissue, cell, or body fluid. That is at primary level of manufacturing. No animal derived materials are used as starting material or in the primary level manufacturing process. The components used to manufacture the raw material ingredients are not derived from animal tissue, cell, or body fluid. That is even at secondary level of manufacturing. C. Ingredients components may be produced by or extracted from plants, bacteria, or yeast, or cell-free systems derived from them, or may be produced by organic synthesis. D. May contain recombinant animal or non-animal proteins provided they are expressed in plants or microorganisms. So the options are only A is true, only B is true, only C and D are true, and A, B, C, D all are true. So it's not exactly very problem oriented. It's just that you have to be very sure to know what exactly an animal origin free medium is, to know which of the statements are true. So an animal origin free medium, you can see here, is not directly derived from animals. Example: salt solutions, basal medium, and complex media. Why we use this? Based on the applications and the advantages of this, you can decide what should be true and what cannot be true in this. So what is the main purpose of this, or what are the major applications for this? Is it is common in vaccine development. So you should know, if you're using this for vaccine development, should it have components from different human, animal, etc., or not. Okay, so then we should also know it reduces the risk of variation. It less regulatory scrutiny. So the ethical regulation or any sort of regulation, less regulatory scrutiny is done for this. Supply is not dependent on the biological products. Certain applications may require additional growth factors or cytokines. So based on all these things, we can decide what components could be appropriate for this media and what components are not. So that is it for this session. So because there are no students in this session, we will uh, end the session. Okay, thank you.